speaker today. So this is Dr. Hannes Dempelwolf. He is the Director of External Affairs and a Senior Scientist at the Crop Trust, where he coordinates the Crop Trust External Affairs team. So he works at the interface of partnerships, resource mobilization, project development, science, and policy. And before that, he had been leading the Crop Trust project, Adapting Agriculture to Climate Change, collecting, protecting, and preparing wild crop relatives. So Hannes studied plant science at the University of Edinburgh and the Royal Botanic Garden, Edinburgh in Scotland. And he completed his PhD in botany at the University of British Columbia, which was a career highlight because that's where we met.
That's right.
It was the best time of his life, I'm sure. He also met his wife there, but that's secondary, I'm sure, to meeting me. And before joining the Crop Trust, he led several field research projects on plant genetic resources in Peru, the Caribbean, Kenya, and Ethiopia. His scientific interests focus on the evolution, maintenance, and conservation of agrobiodiversity, the importance of such diversity for farming communities, and the role it can play for sustainable development and food security in these times of rapidly changing climate. So Hannes, you can get started with your talk, and then I'll help moderate questions at the end. So
great. First of all, thanks a lot for inviting me to be part of your esteemed speaker series. That certainly puts the pressure on. Let's see whether I can garner enough steam to earn that title. The task I was given is to give you a little bit of an introduction of sort of general work we're doing. But I don't know how you handle this usually if people interrupt. I'm very open to interruptions. I think that's a great way to sort of make it interactive, but we can also take questions at the end.
Sure, yeah, we can do both. So if anyone has a burning question, you can do the hand raising icon or just unmute to interrupt. And then we'll take questions at the end as well.
Perfect, that sounds great. Excellent, so thanks again for the invitation. And I'll give you sort of a whirlwind tour of the work that the Crop Trust does, and I hope it inspires some questions later on. So the Crop Trust, who are we as an organization? You may not have heard about us before, which is excusable in some people's minds, not in mine. It is a fairly small organization. We're only about 35 people that work here in our headquarters in Bonn, in Germany. But we always say we're a small organization with a huge, massive, and super important mandate. And that's what I'll be talking about today. So the outline of the talk is the Crop Trust in a nutshell, then what we do in support of ex situ conservation of crop diversity. I'll talk a little bit about our work on global conservation strategies and the Crop Wild Wildlife project that Carolyn had just mentioned. So the Crop Trust in a nutshell, our mission is to develop and sustain a global, rational, and cost-effective system of ex situ conservation of crop diversity by providing sustainable funding for key collections, promoting availability of collections and information sharing, and to fund technical assistance and capacity building. So it's important to understand that we're born out of the UN system, the International Agricultural Research System, the CGIAR, was one of our founders, and so was FAO. And we're really, when it comes down to it, we are a funding mechanism. So we are set up as the funding mechanism whose responsibility is to fund gene banks over the long term. That's when you boil it down to what we are in essence. And what's really important to understand about us is that we are part of a large international framework as it relates to crop diversity, a larger policy framework, and that's the International Treaty on Plant Genetic Resources for Food and Agriculture, which is an important treaty that you should all know about, which I'm sure you probably do, as it relates to biodiversity in our food systems. That treaty is now has more than 140 countries that are party to that treaty, and Canada is actually one of the early signatories who's been part of that treaty for a long time. And most importantly, the US your neighbor next door, also changed that treaty in 2016, which was a big surprise for everyone. It was one of the last actions of the Obama administration at the time. But that means that now most of the countries that have large collections of genetic resources are part of that treaty. And that's really important because this treaty provides the legal basis for exchanging germplasm, exchanging seeds between seed banks and users of seed banks, whether that's breeders, researchers, academics, farmers. This is now all one system, and that system establishes under which rules and regulations that material is shared. And the great thing about a treaty, you may not like treaties and rules and regulations about germplasm sharing. We hear that a lot, but at least it's the same rules that apply across any collection that's in that treaty. And that is now all the CGIAR collections and a lot of all European collections, Canadian collections under federal control, US collections, and so forth. So yes, it's an important part of that treaty, and that treaty recognizes the trust as what they call an essential element of its funding strategy. And I'll talk a little bit later about why this is so important. So in a nutshell, what we do, we raise funds for long-term sustainability of crop collections. We raise the collective profile of collections and their value. We always advocate for the importance of crop diversity and the importance of the conservation of that diversity. And we try to develop high standards of operation through quality management systems. So whenever a gene bank is trying to get funding from the trust, we require for them to meet certain performance targets and to also have quality management systems in place to ensure that the material that we're funding, the conservation that we're funding, is actually valuable in terms of its outcome. So the seeds that are being distributed is viable seed, for example, and it's accessible and it can be distributed. So when we talk about the sort of three-tiered global gene bank system, we talk about the international gene banks, that's the CGIAR gene banks that I had just mentioned. Those are the most important international gene banks around the world, and you probably have heard about them, depending a bit on what you're working on. But there's IRRI, International Rice Research Institute in the Philippines, for example, or CIMMYT in Mexico, you may have heard of, that focuses on wheat and maize. We fund those international gene banks that are attached to these institutions. And then there's several important national gene banks in developing countries that we are also supporting. And we also support and co-manage the Global Backup Facility in Svalbard in northern Norway. So that just gives you an overview of where those CGIAR gene banks, these international gene banks, are located. As I mentioned, all across the world, mainly in developing countries. And they're there to, the mission of the CGIAR is to improve crops and do research for development, so to increase food security in developing countries. The system of CGIAR collections, it's a pretty large system. It has 736,000 accessions, which taken together makes it one of the largest gene banks in the world. And it responds to a lot of requests. There's more than 1,000 requests that come in every year, and they distribute around 80 to 130,000 samples worldwide to more than 100 countries worldwide. The material that comes coming out of the CG collections, so sort of split, half of it stays within the CG and 48%, so a little less than half, goes outside the CG. And the purpose and request for that material is a lot of research and breeding. The international gene banks, here they're listed here, show you which countries they've been distributing in the last few years. And you see that the samples really go everywhere, almost everywhere around the world. And this is a little snapshot to show you the different centers where they are mainly distributing. And you can see that some of the collections are more important regionally. The ICRAF collection, for example, in Nairobi in Kenya, is primarily a collection of fruit trees and other types of agroforestry trees that's active, or that is focused on African and sub-Saharan Africa. And so most of those collections and distribution is in fact in sub-Saharan Africa. But you can see some of the other collections, they're truly used worldwide. So between 2018 and 2020, more than 130,000 samples were distributed outside of the CGIAR, and about 77% of those went to developing countries. If you look at the statistics of what they're used for, what I think it really sticks out is that research purpose is the most stated use of that material. It's not breeding, it's not use in farmers' fields, it's not characterization, evaluation, it's research. And I think this is why it's also so important that the universities and academics pay attention to these collections, because it's actually, in the end, a lot of academics that are using these for various research purposes. Where we did, back in 2019, we did a user survey of the International Rice Research Institute's gene bank to try to figure out what part of their collections are currently used and what part of their collections are most likely more important in the future. And you can see quite interestingly how wild species, wild rices, the demand is likely to increase, and also genetic stocks, so materials that are used for sequencing or sort of pre-bred lines, those types of recombinant inbred lines, those kinds of genetic stocks will be more and more important in the work of the gene bank. Now, I had mentioned earlier that we are a financing mechanism, and I think what's really key about us is that we are trying to fund collections over the long term. So the idea here is that we need an international funder like ourselves that funds these collections and not on a project basis, but over the long term. And the reason here is simple. If you have a project funding for your gene bank, especially in developing countries, if the project funding runs out and you have to switch off your freezers, the electricity supply to your freezers, then within a matter of hours or not days, your collection could be completely destroyed. And that's why it's so important that these collections receive sustainable funding that is not project-based. And the way to do this most effectively is through an endowment fund. And so this is what we've been spending a lot of effort on, is raising the capital in the endowment fund. At the moment, we have an endowment fund of $360 million US, which sounds like a big chunk, and it is a lot of money. But we're still hoping for quite a bit more. Our endowment target is $850 million US. And with that, we believe that we could annually disperse about $34 million US, which would allow us to fund the global system of gene banks in its full extent. We're still a ways away from that, but we're inching closer. As you can see here from the cumulative contributions from that graph, that we've actually had a really good year last year. The market value shot up. The endowment in itself raised, just by being invested in the stock market, $45 million US, which we always say the endowment is its best fundraiser. And we can basically sit back and relax. No, that's not true. We're trying to obviously accelerate the gains made here on the stock market with additional contributions. But we've also been able to use the returns from the endowment. 54 million have been dispersed since 2003. So that's, the mechanism is working. What's important is that the gene banks that receive that money, they have to meet these performance targets I mentioned earlier. And that means 90% of accessions have to be available. 90% of accessions have to be safety duplicated. There has to be a QMS system in place, and we have to have data on the accessions that are conserved. The accessions are no use if we don't know what they are. And with that funding, we provide, we basically provided for all activities that are essential to keep the seed alive. So that's long-term storage, safety duplication, regeneration, characterization, information management, disease testing, and distribution. What we don't fund from the endowment are things like sequencing a collection or evaluating it for drought. That's the kind of funding that we would provide through projects, but not for the long term through the endowment. A few years ago, a big milestone for us was in 2018, when for the first time we funded a full collection of a CGIAR center. That was the first time the endowment had reached a level where we could do that. And it was the Rice Research Institute in the Philippines that has conserves about 130,000 varieties of rice. And we are now able to fund the conservation of those in perpetuity forever, which was it was a big deal for us, because it meant that we as an organization and the mechanism of the endowment fund is working. What's really important is that not only, and I had mentioned this before, it's not only about the germplasm that's conserved, but we also need to take care of the data. We are managing a big information portal that you may have heard of. It's called the Genesis information portal, and that is providing access to information on genetic resources from gene banks anywhere in the world that are participating in the system. And it's quite a lot of gene banks that do that. All of the CG information is in there. The US information is in there. The Canadian Gene Bank is submitting their data. All the European collections are in there. The Australian collections are in there, and many more. So this is a big effort. We have now about 4 million records in that database. And if you are interested in using any accession from any gene bank around the world, I encourage you to check it out. Anything that's georeferenced will show you where in the world it's been collected. Anything that has data on it, passport data, will also show up. So this data is super important with regards to the value of those collections. If we don't have the data, if we don't know what's in the collections, it's basically worthless. This is just to show you all the georeferenced collections that are in Genesis, and you can see that the coverage globally is pretty good. There's a lot of all the main agricultural zones in the world are pretty well collected for a lot of the crops. Another thing we focus on is disaster recovery. We have an emergency grant mechanism where we help gene banks in need, but unfortunately not always are we in time. Not always can we provide the funding that's needed to fix up gene banks that are undergoing a crisis. Here is an example of the Philippine National Gene Bank. It was ransacked by a typhoon in 2006, and then an electrical fire destroyed a large part of their collection in 2012. Unfortunately for that collection, our help came too late, and the other big problem for that collection was that it hadn't been backed up. This is one of the most important activities that we conduct with gene banks around the world to make sure that it's a safety duplication, ideally at 2 levels, but at least at 1 level. And this is also the reason why we have been investing quite heavily, and we've been involved in the foundation and the co-management of the Svalbard Global Seed Vault, which is the ultimate backup facility that you may have heard about. That's a seed vault, so it's not a gene bank as such, but it is a repository for seeds that only other gene banks can use to deposit their backup seeds. It's in the far north of Norway. You can see here on the map the little picture where it is. It's halfway between the mainland of Norway and the North Pole. It was opened back in 2008, and it has the capacity to store about 4.5 million accessions. At the moment, one of the chambers is now contains around 1 million accessions, and is almost full. So they're opening now the second chamber for storage. And this is just a little sort of infographic to show you what it looks like. It's this facility that's built deep inside the mountain, inside of a mountain in the permafrost in the Arctic, and it's really, really cold up there. The permafrost has the natural temperature at about -4 degrees, and then it's artificially cooled down at -18 degrees, which is the optimal seed storage temperature for most crops. Back in 2015, the first retrieval happened from the vaults, and that was because the ICARDA collection in Syria was no longer able to operate due to the civil conflict in Syria. And so the gene bank was basically later ransacked, and a lot of the collections were lost. But they were able to reestablish that seed bank in Morocco and Lebanon by retrieving their material from Svalbard, where they had been wisely storing that material before. And so this is just one example of, I think, what sort of showed the worth of the vault. Why it was important to build that in the first place really became apparent back in 2015. Interestingly, there were so many accessions up in the vault that from ICARDA that had to be withdrawn, that withdrawal process took 2 or 3 years. And then all the material had to be planted out in Morocco and Lebanon, and then the gene banks reestablished, and then backup material be sent up back to the vault. And that's actually a process that's still ongoing with ICARDA redepositing material at every opening. Another thing we've been doing lately with national gene banks is that we've focused on sub-Saharan Africa, and we're supporting 5 national seed collections in 5 different African countries to try to get them up to speed. We're trying to build the capacity of their staff. We're trying to see whether there are any fixes to infrastructure that we need to provide. And we try to sort of bring them into the international system of gene banks in terms of also their policy work and the way they share material. That's a project funded by the German government, and we're very glad to be able to have this support that we can focus on not only international gene banks, but also national ones. Then the third part of my talk, I said I'm going to talk a little bit about our work on conservation strategies and why that matters. What we're doing with global conservation strategies really goes to the core of the global system. If you can imagine, a global system of crop diversity conservation only really works if you know what you're conserving, why you're conserving it, and what your future priority for conservation should be. So we've been doing this for about 15 years now. We've been going crop by crop or crop group by crop group and work with experts to try to figure out what should be conservation priorities. What are the challenges that crops and their genetic resources are facing, and what is it going to look like in the future? What are the priority actions that we should be taking? The strategies generally have 3 components. There is a sort of background on the economic importance, biology, and agronomy of a certain crop. Then we look at we provide an overview of existing conservation initiatives. Where in the world are materials held for certain crops, and where are the most important collections. And then we do a threat assessments. We look at which one of the conservation facilities have the biggest challenges. What are those challenges, and how could we as an international community help to make sure that these crops are conserved properly? One approach that we've trialed since a number of years now that's been quite useful for understanding the diversity in our collections is what we call diversity trees. And this is sort of, it's not strictly based on a phylogenetic tree approach, but this is looking much more at what kind of information, what kind of data do we have available for specific collections, whether that's phenotypic, whether that's genetic, anything goes, even expert knowledge or geographic origin data. And we try to cluster the collection according to what experts think is a reasonable clustering approach. And that has been quite illuminating, I think, because you can do this on a collection basis, but you can also do it on a global basis. And it's a really nice tool to show where big gaps are, taxonomic gaps, geographic gaps, or different trait characteristics that may be missing from a collection globally or in an individual gene bank. And all of the new conservation strategies include these diversity trees. And since I know that there's quite a bit of interest in apple research and breeding at Dalhousie, I thought I wanted to point out that we have done a global strategy for the conservation and use of apple genetic resources, which was done in cooperation with the USDA at Fort Collins. And if you're interested in apples, it's an interesting read, and I encourage you to check it out. It's available from our website. We're also working on a range of other updates of conservation strategies and new strategies, especially in vegetables. So there's a renewed focus there over the next coming years to work with expert communities to develop those strategies. Right, and now the last chapter, the last part of my talk was I wanted to focus a little bit about one specific project that we've been running, and that's the Crop Wild Relative Project. The motivation to work on wild relatives, if you work with gene banks and you sort of start to understand what we have in collections, one thing that really sticks out is how few wild species there are compared to the domesticated cousins of those crops. Yet wild species are actually really, really important for breeding, especially climate change relevant traits. And so we've looked at, can we pull together an international effort to try to collect more diversity, more wild relative diversity from the field, actually utilize it in pre-breeding efforts, and then also make sure that the diversity is conserved in gene banks. And we've done that over the last 10 years. This is actually the last year of the project, which I'm kind of sad about. But on the other hand, all good things have to come to an end. And it was an amazing commitment by the Norwegian government to fund this for 10 years, to the tune of $50 million, which allowed us to work with more than 50 countries around the world. This is just a brief overview of what we did, the different phases. We are now in 2021, which is actually already the project extension. We got an extension for 1 year, and we're just wrapping up all the final activities here. The project has, as I had mentioned, focuses on the main outputs are seeds that have been collected, newly collected wild wildlife seeds that are now available in the terms of the treaty. Pre-bred lines that are now available for anyone who wants to use them, especially relevant for researchers and breeders that are interested in adapting particular crops to climate change. We developed a lot of capacity in collecting on conserving wild wildlife in national programs, and all the data that's been collected throughout the project on the pre-bred line, on the during the collecting missions of the actual wild species and the location data and the passport data and all that is now available publicly online as well. We worked on 26 different crops. They're just an overview is shown here, and you can see there's a lot of important global food security crops are covered by the project. One of the first things we did is try to figure out where in the world these wild relatives have already been collected in the past and where they are collecting gaps. We did a big study about 10 years ago to basically do species distribution modeling on these based on these occurrence data that we had for certain species. And then we compared it to what was in gene banks, and that basically allowed us to figure out where in the world we should be going, or which countries in the world we should be prioritizing for collecting new accessions. And we came out with this map. This was the output of that big global study, which actually shows you that there's a lot of material still to be collected in Europe. Partly that's an artifact of the fact that a lot of our crops originate from Southern Europe, and so there's still just a lot of diversity there. But it also shows clearly that there's still gaps, important gaps that need to be filled. But there's other important gaps around the world, and we tried to target those as best as we could. And you can sort of see the different countries that we went to all around the world in supporting the collecting of those wild species. This is an overview. This slide shows you an overview of the pre-breeding projects that we then kicked off. The pre-breeding projects used wild species to cross them with with domesticated varieties and provide pre-breeding material that then can be taken up by breeders later on. It's a really crucial step when it comes to the utilization of wild species. And we did this on 19 different crops. And we always did it with climate change in mind. So we actually looked at all these different crops and ran some modeling to figure out what are the most relevant traits for each crop to in the context of climate change. We did a lot of expert consultations to figure that out as well. And this is the traits that we landed on are shown on this overview. And for those 19 crops, we then supported projects to actively introgress wild genetic material into domesticated gene pools. And yeah, a big focus here was on abiotic stress related traits, as you can see from the list here. This was done intentionally because wild relatives have been used quite considerably in the past for biotic stress tolerances, for disease resistance, and that kind of thing. And we wanted to push the envelope a bit and look at abiotic stresses primarily through this project, since those are so important in the context of climate change. Drought was a big one, as you can see here. All the pre-breeding projects that had drought stress as a breeding objective are circled here in blue. One example, I'm not going to bore you with the details, but I think visually also very appealing project is our eggplant project, where we work with the University of Valencia, as well as partners in Cote d'Ivoire and in Sri Lanka. And they worked on introgressing more than 14 wild species generated 9 interspecific hybrids, a whole bunch of backcross populations, and introgression lines with different species that they then advanced so far that they could actually look at towards variety release in the not too distant future in both Cote d'Ivoire and Sri Lanka. The Eggplant project was one of the earliest and most successful projects where we could also really work on the data availability. So we've partnered with the James Hutton Institute in Scotland, which is, if you're familiar with, a pretty important agricultural research center in Scotland. They do a lot of work on barley and potato, but they also work a lot on data visualization and trait data and genotypic data and passport data, and in a platform they call Germinate. And we established this relationship with the James Hutton Institute and have now worked, I think, on 11 different Germinate instances. So those are the Germinate tools applied to 11 different crops to be able to share all the breeding relevant information, all the sequencing that's been done, all the genotyping that's been done, all the phenotyping that's been done in one accessible database that's clearly linked back to the germplasm that is also available. I think this is something that a lot of research groups struggle with, on how to manage your data and how to make it accessible to users. And I can really recommend you check out the Germinate solution that the James Hutton Institute has been providing and developing. We've had a lot of luck with them, and we've had a lot of good feedback as well. This is just a screenshot, basically, on what it looks like when you're in the when you're looking at that at those data sets. And the last a bit of promotion that I wanted to put in here is we just had published a special issue of Crop Science, which highlighted a lot of the outcomes of the pre-breeding projects of that we have been supporting. I think almost all of the papers in that special issue were a direct outcome of the funding we had provided to utilize wild relatives for climate change adaptation of different crops. So if you're interested in learning more about that, I encourage you to check out that special issue in Crop Science that was just published a few months ago. And with that, oh yeah, now I had one last slide sort of summarizing the outcomes of the Wild Relative project. And just to go through it again, we had more than 4,000 new collections, working with 62 partners in 34 countries. And there's been lots of good material that's been coming out of those pre-breeding projects. Kathleen, actually, when she asked me to give this talk, she said I should really end with a call to action. So today I thought about what could that be for you guys, and I decided to talk a little bit about one issue that's been on my mind for quite a while now, and that I think research and academics really have to pay more attention to. And this is also, I myself come out of a Canadian university, as Carolyn had mentioned, and I know about this issue, that there's actually a lot of genetic resource collections held by labs as research collections. And those are often extremely poorly documented, very little thought given to long-term conservation. There's no mechanism for sharing according to agreed international rules. There's often a considerable risk of destruction or complete loss of collections if professors retire. We've had we actually getting quite frequently requests from universities saying, do you want those 5,000 barley accessions, because otherwise we're going to burn them if you don't take them. And that is, to me, those are high crimes if you really go through with that. And we know of cases where in fact whole collections have been destroyed because they didn't want to deal with the IP issues around figuring out how to share the material or get it conserved. And so there's often also no national solutions that could take on those materials or the conservation of genetic stocks or sequencing sets. It's a real big gap in the whole system. We work a lot with national collections, but university collections don't fall under the same rules. They're often not part of the federal government, which I think is also true for Canada. They're often provincial facilities. And therefore, different rules apply, and provinces and provincial governments often don't pay attention to these issues. And this is something that we're hoping that the academic community really starts paying attention to more. When there are collections, and a lot of labs have these collections, have you thought about backing up valuable material at gene banks? Have you thought about what happens to the collection once the important parts of our lab retires, or if someone moves? What does the university say about these collections? Is there any sort of commitment to continue to conserve them or make them available? And I think we should also, as academics, should ask funders to support the conservation or the availability of that material when you ask for funding for your next project. You should think about what's going to be my plan at the end of the project in terms of making the material available. Can I partner already gene banks now to see whether they have an interest in taking it? How am I going to fund that? Is there a way that we can ask the donors to actually contribute to that? I think these are all really, really important questions that every researcher who is thinking about developing a project around germplasm improvement or even diversity research should be asking themselves. So I hope this is enough of a nagging call for action here at the end. And I thank you very much for your attention.
Awesome talk, Hannes. Thank you so much.
Sure.
So if anyone has questions, we'll open up the floor to questions. You can, oh, Paul's giving the little applause. Yes, we can't, if you were in a room with us, we would all clap very loudly, I know. So if anyone has a question, you can turn on your mic or raise your hand.
Or show your face, God forbid.
Turn on your camera if you'd like. Go ahead, Paul.
Hey, thanks so much, Hannes. That was awesome. Can you hear me okay?
Sure. Yes, I hear you perfectly.
Perfect. Okay, so I'm not a plant geneticist by any means, so this might be a really terrible question. But when you're showing those different species and all the 21 species in your recent project and looking at all the different climate-related adaptation or climate adaptation-related traits, and sunflower had time to flowering for one of the traits. And I was thinking just about, at least in our region, we're having these issues with these early frosts and both at harvest and at early stages. So is that a major target across other species to get that plant from seed to fruit or seed to stalk or whatever it happens to be? Is that a widespread trait? Or is it mostly environment related rather than gene related? Sorry.
It really depends on the species. It's always the best answer is to say it depends, but it really does depend. There are some crops where this is also related to drought avoidance. If late in the season you get big droughts, then there's a big advantage of going to fruit quickly. And in that context, it is a trait that's of relevance, also in terms of climate change related traits. But I have to say, the sort of Canadian perspective is not something that we've taken strongly into consideration for this project, simply because our focus on developing countries and cold tolerance is not generally one of the biggest stresses that is being encountered.
Cheers. Thanks so much.
Sure.
Thanks, Paul. Yeah, I was thinking similar things because climate change models for our region, I think, actually predict us to get wetter. But yes, we are not a developing country, so probably not the target.
But wetter soils is definitely an issue. Waterlogging is a problem also in developing countries. You also have issues around coastal areas and sea level rise, which has similar problems. Of course, that's often combined with salinity. But our carrot pre-breeding project actually looked at, if I remember right, at waterlogging also as one of the issues.
Cool. Okay, I had a question as well. So earlier on in your talk, you showed what the withdrawals from the CGIAR banks were used for. And in 2020, there was a huge jump in direct use. Do you know what caused that? Or did you guys have any kind of campaign to encourage direct use? Why did it go up so dramatically in 1 year?
Yeah, no, it's an interesting observation, and quite frankly, I don't have an explanation. I can speculate though, which I like to do. And so sometimes, as you say, sometimes there's campaigns that are run by either the CG centers themselves, where they try to actively engage farmers in direct use of their collections. when it's a big jump like that and it goes down the next year, generally that's the cause. That there's been a big global project that has been using material. If it's a more systemic trend, then you would see it sort of creep up over the years. And so I would attribute that probably not to a real sort of demand-driven distribution, but rather a supply side driven distribution because there's been a project that encouraged to use.
Thanks. Any more questions?
Okay.
I have a quick one, if that's okay.
Sure.
Thanks for the great presentation. It was really interesting to hear more about what the Crop Trust does. I was just wondering, like at the end of the CWR project, what were kind of the remaining challenges to like protecting crop relatives? Is it to do more collection, or is it what kind of is left? And now that that project's over, is there plans to continue on kind of that concerted work?
Yeah, that's a very, very, very good question, and there's a lot of answers to that question. So, firstly, it's incredibly time and money consuming to do collecting of wild species. And I think we had started out thinking that the kind of investment we did, which was truly global with lots of countries, would probably get us more than the 4,500 accessions that we did get. If we did rerun the gap analysis, and there has been a decrease in the gaps, but there's certainly a lot of gaps remaining. So further collecting is absolutely continues to be a priority. But I'm not sure you can actually pull this off as a global project. I think there needs to be a lot more national investment, and so it's a long-term thing. You have to do this, go out every year and try to hit your collecting spots to really sustainably increase. So that's one issue. Another issue that still remains to be supported is the conservation issue. So collecting wild relatives is one thing, but conserving them is actually also really, really tricky. These wild species, we don't even understand how the biology works. How seed set works, how we regenerate them, how we germinate them. There's a lot of basic questions that are not known for a lot of these wild species, and we really need to invest into doing some research around figuring that out, helping gene banks understand how to better conserve their wild species, and also financing them to do that. I think that's another big issue. A third issue, I would say, is the sort of last mile problem. We've developed a lot of these pre-bred materials, and these pre-bred pools are now available. But what's the next steps? How can we encourage the use of that material and inclusion of that material in breeding programs? And I think a lot of that, depending on the crop, the private sector is important. Public-private partnerships around utilization of that material is probably something that should be looked at. And we ourselves we've started working directly with farming communities in these pre-breeding projects, which in itself is a weird concept because the pre-bred material is not released varieties, and it's kind of, it's still a few years away from something that a farmer would usually look at. But we've had some very encouraging and positive experiences where farming communities were able to identify really valuable material straight out of the pre-breeding pools, and were wanting to use that and multiply it. So that's one of the sort of more researchy trajectories that we're trying to encourage in a follow-up project, to see to what extent can we move pre-breeding and farming in a participatory pre-breeding in some ways. How can we advance that? So those are some of the priorities that are still left to do on wild relatives.
Cool, great, thank you.
Sure, welcome.
Raf, you had a question? You want to go ahead?
Yeah. So thank you very much. I really enjoyed your presentation, but I was thinking about how do you, I believe through your collections, you have some recalcitrant seeds. And so how do you maintain these ones over time? Because like not all seeds can be stored in cold places. So I don't know if you have got some field where you constantly regenerate these, and how do we get access if you want to have, let's say, like use some of these plantlet for research purposes?
Yeah, excellent question. And recalcitrant sheets are a real headache for us who work on GMAX. And the answer is, as you had mentioned, so there's either field collections, which are inherently vulnerable. Field collections are collections in the field, and if you have a major environmental stress or a disaster happening, then your field collection may be wiped out. So they're not a great way of conserving over the long term. So what we've been investing in quite heavily is cryoconservation, where you introduce some of those plantlets into cryogenic storage at -80 degrees. Really, really expensive to do that. Also very difficult to do that for different species. Every species has their own cryogenic requirements, and how to introduce that can be very, very tricky. Really labor intensive also. But once you have them in cryogenic storage, then it's anyone's guess how long they survive. I mean, they're potentially in there forever. So it's a very safe way of storing material, but it's expensive to introduce these materials into cryogenic storage. But then there's also the crops. Clonally propagated crops are often kept in vitro collections. Potato is a good example, or bananas, where they're actually grown as little plantlets, and they're being grown over and over again in vitro cultures. Also incredibly expensive to do that, especially if you're talking about thousands of collections. Within the CGIAR system, the most expensive gene bank is probably the potato gene bank in Peru, and they only have about, I think, 8,000 accessions. If you compare that to the rice gene bank, which is at 130,000, it's a drop in the bucket. They cost at least 3 times as much to maintain the potato collection, simply because it's so expensive to keep these in vitro cultures going. So yeah, I hope that answers a little bit your question.
Yeah. Thank you. Yeah, but I cannot follow up. Yeah. So, for instance, if I need some genetic material for some research purposes in terms of abiotic stresses, which is your main focus, like I would like to know how can I get these, and is there any approach where maybe I need to contact some people, or for instance, I'm a young researcher, I tend not to have enough funding. How do I collaborate, or how can you come on board to help with this research?
I mean, so as a matter of principle, all the material in the collections that I've been describing are free of charge, and that's due to international law, and because we exist, because we fund these collections. So the collections themselves cannot make money of supplying material to you. What they may charge you is shipping costs, but they will not. They cannot. They're not allowed to sell their material. So generally, it's cheap to get material. The way you figure out how to get material is a good way to start is to look at some of these databases. And the biggest database I had mentioned is the one that we're managing called Genesis. I'm just putting the URL here in the chat box. That is a good entry point to try to figure out where your species of interest, in which gene bank it's located. Once you figure that out, there's ways to, depending on the gene bank, you can try to file an order through the Genesis system, but often you have to contact the email, the gene bank directly. But you'll find the contact information and the details on how to do that in the Genesis system. And if you fail to do that, then just pop me an email, and I'll try to put you in touch directly with whoever you're trying to reach.
Okay. Thank you.
Sure, you're welcome.
Paul?
Yeah, this isn't quite a scientific question, but just a planty question. I remember a couple of years ago seeing this really amazing landrace of corn that had these like aerial roots with all this mucus that had nitrogen fixing bacteria.
I remember that.
Did you find anything really fascinating like that with this big crop wild relative search? Anything that came out with really interesting traits, or yeah, I'd love to hear more about some cool plants.
Yeah, I mean, it's probably too early to find to talk about the traits of the newly collected material, because the collections is just finished, and there's a lot of evaluation characterization work that has to be going on now to figure out what the traits of interest are. But there were several populations that were found of material that thought that had gone extinct, which is interesting. And in several countries, we also encountered material that we thought would be abundant that was almost didn't exist, and sometimes it did in fact was never found during the collection trips. That's probably some of the most to me, some of the most shocking revelations that are part of that project, the stuff that you can't find anymore because it doesn't exist for whatever reason. Yeah, so I think in terms of cool planty stories, I would have to dive a bit more into the archives. I don't think any mucousy aerial roots come to mind. There's some cool ones that I thought were that look really cool and creepy are spiny eggplant wildrolifs. They have these crazy spines on their leaves, and you cannot imagine how someone thought this would be a great thing to cultivate and domesticate, but it clearly happened. And so some of these things are cool to see. Yeah.
Have you ever been to the seed bank in Svalbard?
Yeah, I've been there several times.
So cool.
Yeah, no, it's an amazing experience to go there and go into that mountain. It's also really, really cold. Generally, the best time to go there is actually in February, when it's the end of the Arctic night, when the sun sort of comes back a little bit, and it's basically from in the morning you wake up and you have this beautiful sunrise, and then it slowly goes into a sunset, and it's basically the whole day, the whole sky is on fire, and then when it's night you have the polar lights going up, and it's just so bitterly cold, and you have the reindeer stalking around everywhere. It's like no other place I've been to.
So how do you get there? Like, do they have an airport?
Yeah, it's actually amazingly easy to get there.